Hey everybody, Tom and I are here at Your Amigos Live 2025, the fourth annual, believe it or not, and we're going to do a video on VEGF, PD-1, PD-L1 bispecifics. This is an exciting new class of drugs, and as the name would imply, this is a one molecule bispecific antibody, targets VEGF, targets PD-1 or PD-L1, depending on the molecule. My understanding of the preclinical data is that it's not just like two drugs separately, that when you bring these binding moieties together, you get cooperative binding. So you get better VEGF binding, you get better PD-1, PD-L1 binding, and you get enhanced effects on each of those pathways.
My reading of it is that it seems to be the VEGF close location to the PD-1. The topography seems to be potentially that more of an immune stimulant than a VEGF-targeted therapy. You might remember Dave McDermott's study with bevacizumab and PD-1 back in the day.
Remember
that study where there was discussion about T-cell trafficking. And the feeling is by having these two proximity so close to each other, you can really facilitate that T-cell trafficking.
Yes.
There is quite a lot of preclinical data to support that, but also on the negative side, you know, if it's not different from VEGF PD-1, we've done lots of those experiments already.
Agreed.
And so the question is there any clinical data out there to suggest it's better than the two that we would expect? And the answer to that question is there's a drug called ivonescimab, and this is a PD-1 VEGF bispecific, and this was tested maybe 18 months ago, a randomized phase 3 in lung cancer, and in non-small cell lung cancer, it showed better PFS, significantly better PFS than pembrolizumab, which, you know, everyone thought that's been really hard to beat. You remember pembrolizumab had fabulous activity in non-small cell lung cancer. That made everyone sit up, and that's launched a whole string of other drugs that you're going to talk about in a second. But there are 4 or 5 being developed, including a PD-1 VEGF bispecific. I'm going to give you an update on that data, that ivonescimab data in lung cancer. So they didn't hit overall survival in that randomized trial. And that happened at World Lung this summer. There was some issue about some subgroups, China versus rest of the world. And it wasn't a huge trial. It was a bit underpowered. I'm not surprised it didn't hit. It trended the right way. It was in the 0.8s, but people were disappointed by that. But then at ESMO, there was another trial, this time in combination with chemotherapy. So ivonescimab chemotherapy in non-small cell lung cancer versus chemotherapy pembrolizumab. And again, it beat pembrolizumab for PFS. We didn't see any OS at this point. I mean, it was positive. I think we would have seen it. But anyway, the point is that's 2 trials now. So it's not a black swan event. And Brian, you know some data in some other areas too.
I do.
You do.
I do. You love saying black swan event. I've noticed.
Well,
anyway, so BMS has a drug called pumitamig.
Yes.
That is a VEGF PD-L1 bispecific. It was in-licensed from a China company, I believe. And our friend and colleague Zinan Shang presented data at ESMO, I believe, last year, 2024.
Yes.
Monotherapy in a refractory clear cell population and a frontline non-clear cell population. I don't remember if it was just papillary or not. 25 or 30 patients. The clear cell was mostly just VEGF refractory, not so much IO refractory, but otherwise pretty typical of refractory populations, and clear cell, non-clear cell. And response rates in the 25 to 30% range and a pretty impressive waterfall. So to me, the big clinical question when I first heard of this class of drugs, as you alluded to, is it any different than just giving the drug separately? We've done that all day in kidney cancer. It's a little more activity than I would suspect, you know, of this bispecific, but small trial, jury's still out.
And if the VEGF is not as active as a VEGF TKI like cabozantinib, if that component is not as active, it's going to struggle in kidney cancer, isn't it? Because len-pen or axi-pembro is going to get that very powerful VEGF signal, and the bevacizumab signal hasn't been so strong.
Correct.
And so how do you overcome that hurdle? How do you beat?
It better be a really good immune therapy.
Yes.
Because it's probably a weaker VEGF therapy.
What if you combine it? What if you combine it?
Well, so I think the question is, how would you develop this, right? We already have VEGF PD-1 combinations in kidney cancer, a lot of them. So my opinion could be monotherapy in a refractory setting. Maybe, maybe not, but you can go up against belzutifan or something, or maybe more exciting or certainly bigger combination. CTLA-4-based combinations, belzutifan-based combinations, TKI combinations, although it'd be nice to not to have to use TKIs. So that'd probably be my least favorite. But, you know, frontline combinations, single agent refractory. There is an MD Anderson investigator-initiated study. It's single agent ivonescimab in, I believe it's an IO refractory cohort and an IO TKI refractory cohort, if I have that right. Single agent, just signal of activity, maybe it's 30 patients or something.
And then, of course, you could say, why not go after everywhere where pembrolizumab is active?
Let's do it.
And so you could say, well, what about in head and neck cancer? What about in bladder cancer?
For sure.
You could do an adjuvant study in bladder cancer where nivolumab as a single agent is used. Or, as you suggested, you could do combinations with ADCs too. And you could maybe try and find a way of improving current activity.
What would you do in bladder cancer? You can do one setting.
Oh, it's so difficult.
Or prioritize the settings, perhaps.
I don't think as a monotherapy, the one thing we haven't talked about is sequencing.
Yeah.
So and there was some data in kidney cancer there, is if cancers have progressed after Pembro, can you rescue them with this? But my feeling is that's not as exciting as going frontline.
Frontline metastatic.
So frontline metastatic or perioperative, neoadjuvant EV Pembro versus EV Pembro plus this.
Okay.
A PD-1 VEGF bispecific. So that would be attractive. Or, other drug companies could look at the other partnering ADCs. So you could look, for example, TDXD, AstraZeneca, if they had one, TDXD plus a VEGF PD-1 bispecific in heavily pretreated patients. So I think it's a combination strategy. And in bladder cancer, we're using combinations with ADC. So that makes sense to me.
And there's a lot of different of these bispecifics. So you can imagine different partners, et cetera, that are probably all of what we said is going to happen in different settings. But I think it's a pretty exciting new class of molecules.
It's probably caused the most stir that I've seen for some time across the cancer.
Do you know what I don't think I've mentioned?
Tell me.
We're by the Batman building.
Oh, yeah.
Batman building in Nashville. So the Batman building is Batman and Robin, this great duo. And we're talking about this dual drug of bispecifics.
Brilliant for the end, Brian.
Thank you.
Brilliant.
Thank you very much. Thank you.